The Queensland Mycological Society acknowledges the traditional custodians of country throughout Australia and recognises the continuing connection to lands, waters, fungi and communities. We pay our respect to Aboriginal and Torres Strait Islander cultures and the elders past, present and emerging. As I'm speaking to you from the Sunshine Coast, I would like to acknowledge the Gubbi Gubbi people in particular, who are the traditional custodians of this land. Thanks everyone for coming tonight. Tonight we have a very special guest, Patrick Leonard, and I'm going to hand over to Patrick Leonard just now, and he will talk to us on his pet species, his focus group, which is Russulaceae.
This is a talk about russulas, and Wayne asked me to do it at, I don't know, medium sort of notice. And I was going to talk about the russulas we found on a foray at the Annie Hehir Reserve, where we collected 14 russulas a couple of Saturdays ago. And I decided that in order to make sense of it, I really needed to say a little bit about the history of the genus Russula and how people used to actually identify russulas and how that's changed over the years. So I'll do a little bit of history first, and I'll talk about the characters that have been used for identifying russulas, and then I'll get on to what we found at the foray. So let's see whether this works. The Russulaceae were named by Christian Persoon. I think of him as a Dutchman. He was actually born in South Africa. And that was in 1796. And Persoon's concept of the Russula was of a very fleshy fungus with a depressed cap and equal gills. And if I go back to the introduction, you'll see here Russula emetica, which is the type species for the genus Russula. And as you can see, it's got a depressed cap, it's fairly fleshy, and by equal gills, he means that the gills run all the way from the edge of the cap down to the stipe. And there's no, we'll come to later, short gills. So Persoon introduced us to the concept, and then Elias Fries, who's considered to be the father of mycology, expanded the definition in 1825. Fries, of course, introduced the whole concept of using spore colours to classify the fungi, and to this day we still use spore colours to a large extent to separate one genus from another. Russula is in the family Russulaceae, and in the 19th century, Russulaceae had two genera, which were very similar. One was Lactarius and the other was Russula. And you could tell the difference between the two. If you cut the fungus on the gills and it yielded a latex, a milk, then it was a Lactarius. If it didn't, it was a Russula. Life was very simple in those days. We move on to 1900, and a bloke by the name of Mordecai Cooke published a handbook of Australian fungi. That was in 1892, and it really was the first comprehensive look at fungi. Now, Mordecai Cooke trained as an apprentice draper. He didn't have a scientific background exactly, and he became a school teacher, and then he became head mycologist at Kew. He never visited Australia, so he basically worked on the samples that had been sent by Bailey and von Mueller and other people who'd collected fungi in Australia, sent them to Kew, and the handbook described 2,079 species of Australian fungi. Only 10 of those were russulas. Of the 10, 9 had European names. So Mordecai Cooke had matched them to a European Russula that was in the herbarium at Kew. And only one was a native Russula, Russula australiensis. And here's a picture of it from Cooke's book, a painting. It's number 41. It's the big red job on the left of my screen. We'll come back to Russula australiensis. It's not on our current list of finds in Queensland, but you never know. So the key characters used by Cooke were taste. By taste, basically he meant, was it hot like a Madras curry, or was it mild? Over the years, there's been intermediate categories added to those two, but that was what Cooke was looking at. He recorded cat colour, but it wasn't with reference to a colour chart, so you had to interpret his words. He recorded the diameter of the cap in inches, the stipe shape and colour, how the gills were attached and what colour they were, and the spore size. And he did spore sizes in terms of either a single measurement, from which you could assume that the spore was globose, was round, or sometimes he gave two measurements. The whole of Cooke's description for a Russula amounted to about 30 to 40 words, so 5 lines, very different to the descriptions that we have nowadays. We move on about 30 years, and a Czechoslovak medical doctor by the name of Vaclav Melzer made an interesting discovery which changed the whole way we looked at Russulas. He developed a stain which was based on iodine solutions, and he was interested in mycology. He put some spores into the solution and discovered that suddenly the ornamentation that was on Russula spores could be seen much more clearly than it had been previously. So here's an example of the change that happened. You look at the top left-hand corner, that's what Mordecai Cooke would have seen under his microscope as a Russula spore. You can see a slightly roughened edge to it, but no more than that. If you look at the spore on the right, you can see that some Russula spores have warts. They're they've got warts and no other ornamentation. Go to the bottom left. Many Russulas have warts and they have short lines, which are called connectives. And then go to the one on the right. Some of them have like a network over them, which we call reticulation. And suddenly there were these additional things that you could use to identify a species of Russula. We move on to Australia, and in 1934-35, Cleland published his mushrooms and other larger fungi of South Australia. It had 12 species of Russulas in them. In the 1930s, there was still a commitment to the idea that some Russulas were global, and therefore Cleland retained 6 European names for Russulas. None of those names matched any of the European names, the 9, that were in Mordecai Cooke nearly 40 years before. Cleland had 6 native species, and none of the native species matched Cooke's Russula australiensis. So we've got 2 books separated by about 40 years, and both with about the same number of Russulas, but there is no correspondence between them whatsoever. The one change that Cleland introduced into the characters that you needed to identify a russula was changes in flesh color. Some russulas, when you bruise them, go brown or black or red. We move on to post-war, and the leading authority on russulas in Europe, in France, I should say, was Henri Romagnesi. He was a Latin teacher in a lycée, a secondary school, but he spent all his spare time on mycology. And in 1967, he published a monograph on russulas. There were 170 species. His descriptions were rather longer than those used by Cooke. The book ran to nearly 1,000 pages, and it focused on France. Romagnesi didn't travel widely. He went to Britain, he went to Holland and Belgium, but he didn't travel to Scandinavia, as far as I know, or south to the Mediterranean. So his is very much a view of the temperate Russula funga of Europe. His big innovation was that he introduced a spore color chart, and Romagnesi subdivided Russula into a number of subgenera, and the basis of the subdivision is entirely on spore color. Here's Romagnesi's color chart, and you see he's got 4 main classes on the left. The first 2 are basically white or off-white. The next 3 going down are pale cream. And then he has 2 ochraceous spores and 3 what he calls xanthospore, which are almost orange. They're dark ochraceous. So that allowed us to have an extra character to use in classifying and identifying russulas. So following on Romagnesi, we now have these characters which are used to identify russulas. So you look at the gills and you see how they're attached and what color they are, which was done by the previous authors. But he also mentions the presence or absence of short gills, lamellulae. He uses spore print color, he uses taste, and he's got hot, mild, acrid, and fetid as his categories. Cap color and diameter remain, stipe shape and color remain, but now we have spore size and ornamentation. If I go back to Cleland, I don't think Cleland was aware of Melzer's work because he doesn't mention ornamentation in any detail. And in his glossary of terms, he doesn't talk about the amyloid and dextrinoid reactions which accompany use of Melzer's. Romagnesi also introduced the structure of the pileus of the cap and the presence of dermatocystidia. Cystidia are sterile cells that occur on the gill, but they also sometimes in russulas occur on the cap. And Romagnesi used a stain called sulfovanillin, which is basically concentrated sulfuric acid with a few vanilla crystals added to it. It's very good for identifying dermatocystidia and very bad for your microscope. We move forward again to 1997, and Cheryl Gruginovich revised Cleland's collections, which are in the herbarium in Adelaide, the herbarium of South Australia. And she published a book, The Larger Fungi of South Australia, that contained 13 Russula species. All of Cleland's native species are retained, and most of Cleland's European names are now recognized as distinct species and given Australian names. But she also separated out some of Cleland's collections that had either been wrongly named or not named at all, and they're part of the 13. We move on to modern times, as it were, and Teresa Lebel adds a whole lot of new Russula species in a paper published in 2007. And it really marks the entry of Australian Russulas into the whole world of molecular genetics, DNA, and sequencing. Of the new Russulas, 3 were secotoid species that had previously been in the genus Macowanites, and Teresa moved them to Russula. Secotoid species are species where the cap doesn't open properly. They've still got gills inside, but the cap remains closed around the stem. We'll see one a bit later on. And then there were 7 gasteroid species, that's truffle-like species, that don't have gills inside. They have more of a sponge-like material, rather like in a bolete. And they were also transferred to Russula. So by the time we're ready to go on our foray at Annie Hehir, we have 13 agaricoid Russula species. That's Russula species that have a cap and a stipe, and they open up and you can see the gills. All of those have been found in Queensland. We have 3 secotoid species where the cap doesn't open, and only one of those has been found in Queensland. That's Russula Species. And we have the truffle-like species, 4 of which have been found in Queensland. So here we are, ready to go on our foray at Annie Hehir, and we have an expectation of being able to find, well, there's 18 possibilities altogether, I suppose, if you count all the agaricoid and all the secotoid species. But realistically, there are only 14 species of Russula that we might expect to find. So off we go on the foray. There were 11 forayers. No, there were 12. There's one that's not in the photo. And my objectives in going on this foray were to collect at least 2 named Russulas species to have a look at, and to collect at least 2 named bacteria species. I made a resolution not to take home any amanitas. Some of you heard what Fran had to say earlier about identifying amanitas. There's an awful lot of them in Queensland, and they're mostly not in any books. I also resolved not to fall over, not to be bitten by leeches, and not to end up with more than 6 collections to identify. Some hope. Here are the forayers. You can't see Gretchen because she went on ahead of us, having got bored talking to us while we were waiting for the last members to arrive. You can't see Wayne because he was taking the photo. So we found a lot of Russulas. There were 14 found altogether. We don't have photos of all of them. They appear to consist of 11 separate species. And I found that quite remarkable because I haven't been finding any Russulas this season. The first one you see here has a greenish, olive greenish tinge to the cap, and purple on the outside and purple on the stem. This is a Russula that the Queensland Mycological Society have collected before. The first time we collected it was up in the Girrawheen National Park at the underground trail. Anyway, in Girrawheen. And it was written up, and we've found it 2 or 3 times since, but this Russula has not got a name. It's not been published. This is Russula galbana, and you can see what I mean about a secotoid Russula. It keeps its cap closed, and because a hole's been punched in it, you can see that it has gills inside. So it's just like the other Russulas, but a different shape and doesn't open. One must assume that the secotoid Russulas are in some way being spread by marsupials eating them, perhaps, in the same way that the truffle ones are. Third one is Russulaceae, which I haven't seen that often. It's not that common. It's not as common as some of the other ones we're going to see. It's a brown Russula. It's fairly large. It seems to be coming up in several places this year. Prior to this year, I hadn't seen it for 4 or 5 years. So not that common. This Russula also hasn't got a name. It's a sort of pillar box red color. It's got a white stem. This has the original description of gills that run all the way from the edge of the stipe, from the edge of the cap to the stipe, so equal gills. This is Russula neremia, which is very common. Russula neremia is recognized by its smell, something you can't actually show other than by handing the fungus around. Not many people in Australia, I find, know Jerusalem artichokes as a vegetable. But if you do, and if you make soup out of them, you've got the smell of Russula neremia. If you don't know Jerusalem artichokes, they're in season, and if you go to a good greengrocer's, you'll be able to buy some. So Russula neremia is identified by its smell, but it also has this very striate edge to the cap, and it tends to fall apart rather easily. It's a not very robust Russula. I've put up the name of Russula Clelandiae, and if you go on the ALA and look at Russula Clelandiae photographs, you'll see some that are this color. The original description of Russula Clelandiae, which I think was done by OK Miller and Hilton over in Western Australia, describes a much more purple fruiting body and purple stipe. And there's a wonderful illustration of it in Katie Syme's book. Whether the red ones are the same species exactly, I somehow doubt, but no one's done the work yet, so we don't know. There may actually be 3 versions of what is called Russula Clelandiae in the ALA and on iNaturalist. I've put this down as Russula viridis. It's not a good example. It has the olive green tones in the center here, but viridis normally has green over the whole of the cap, and the top layer of the cap in viridis has a tendency to break up and leave patches of the cap and little holes, and it's described as areolate. If you look at the Russula that's behind my head on the video, then you will see a true example of viridis. Some Russula colors are water soluble, and you tend to lose them. And by this stage in the foray, we were experiencing very, very heavy rain and being attacked by leeches and goodness knows what. This is another little red Russula, quite small, always small, fairly short stem, which is about the same height as the cap is wide, dark red, always has ochraceous stains on it. Undescribed, don't find it very often, but red Russulas seem to be very common in Queensland. This is a pink one. Russula nusaensis is an unpublished name as yet. Pink cap, pink stipe, very common in southeast Queensland. I suspect it's quite common elsewhere. I've seen it down in New South Wales as well. It differs from the others largely in microscopic features. And then finally, another pink Russula, this type with a very dark center and white stipe. Again, undescribed, we don't know what it is. So, having seen all these, I looked back at the sort of information that you now need in order to identify a Russula. And it's changed dramatically from those early days of Mordecai Cooke. The macro characters, the ones that you record in the field or when you get it home, are the fruit body form. So now we've got agaricoid, secotoid, and gasteroid forms of Russulas. The cap color, texture, and size more or less remain the same as they have for the last century and a bit. And likewise, the stipe, shape, color, and size. Gill attachment is now accompanied by an estimate of how dense the gills are. There are some with very widely spaced gills, some with moderately spaced gills, and some with crowded gills. Some people use measurements, but there is no standard used with Russulas to separate those three. Colors are done by reference to a color chart. Some people use Romaniesi's color chart. There's a very good Russula color chart in the, I think it's volume 6 of Fungi of Switzerland, which is a rather more subtle division than Romaniesi's one. We now have to see whether the gills fork or not, whether they're furcate, and the presence or absence of lamellulae. A chemical reaction is now widely used in Russulas. You either apply ferrous sulfate in a liquid form, or you use a ferric alum crystal to rub the stem, the stipe of the Russula, and it can turn green or do nothing or turn a salmon color. Important for separating out the Phaetens group of Russulas. And you need to record the associated organism. Extremely difficult in Australia, as Nigel is fond of telling us all, because we have such mixed forests, but all Russulas are associated, they're mycorrhizal, they have a symbiotic partner, and in Europe and North America, where you find forests that tend to be dominated by a single species of tree, and they've identified close associations between the tree it's growing with and the species of Russula. There are some that will grow with more than one species. Certainly the ones that grow with conifers will also grow with birch trees, but for many of the ones growing with broadleaf trees, they have a preferred partner. It would be nice for us to be able to do the same. Microscopic characters have become more detailed. Spore shape, ornamentation, and spore size are all used. Basidia shape and size, and the number of sterigmata, the little posts at the top of the basidia on which the spores are held, are counted. There are some russulas that have a certain number of two-spored basidia, and that causes two-spored basidia in general tend to produce bigger spores. So it causes a variation in spore size. Pleurocystidia, that's the cystidia that on the side of the gill. You note their presence, their shape. They come in all sorts of shapes, and the tops of some of them can be pointed, or they can be moniliform is the word, like the spire of a pagoda. There are various possibilities. And chylocystidia, which are the cystidia on the bottom of the gill, facing the ground, likewise. Dermatocystidia, the things that Romaniesi introduced us to, you record their presence, their shape, and their size. And the piliopellis structure, that's the structure of the top layer of the cap, is also something that is now routinely recorded and put into descriptions of Russula species. It's telling you something about the nature of the cap that you observe. So the Russulas with shiny caps have a piliopellis structure that has hyphae that lay more or less flat on the cap. They're intertwined, but they're more or less flat. Others have hyphae that stick up and hyphae that are gelatinized and so on. So now we have a third category of characters that are recorded. These are DNA, molecular characters of fungi, and we've got to the position, not with Australian russulas yet, but certainly with some other genera, where we can recognize that there are species that we can't actually see any differences of in the field. So my conclusions, well, I met all my objectives, except the ones about leeches and the number of collections I took home. It's difficult to do that. Annie Hehir Reserve is a very good site for russulas. We found a remarkable number. Identifying russulas has become harder over the years, and there's certain things that as an amateur mycologist you can do, and most of the russulas that are so far named in Australia you can identify as an amateur. You don't have to actually go to the DNA of the end of things. But rather like we were saying about amanitas, there are an awful lot of russulas in Queensland. I reckon on something like 60% of them are as yet unnamed. So we've probably got 50 agaricoid ones in Queensland alone. So there's a long way to go, and you'll have a lot of disappointments of collecting a russula and never being able to put a name to it. Writing up the unknowns will help, and sharing it with other members of the QMS so that we can begin to accumulate some understanding of which are the common ones and which not. And I think it would help all of us, QMS, the mycological community in general, if we were training up more identifiers. I don't mean more taxonomists. I mean people who, like Romaniesi, have devoted a large number of years to becoming expert in a particular genus and then are able to do the sort of confirmation job that is needed for people who, the citizen scientists, who are submitting specimens to our naturalists or to the ALA. That's it. That's all I've got to say. Stop share.
Well, thank you, Patrick. That was wonderful, as always. Do we have any questions? So I'll invite everyone to unmute if they wish.
Yep, I have a question. It's Vanessa.
Go ahead, Vanessa.
Hello, Vanessa.
Hi, Pat. That was a brilliant talk, as always. I really enjoyed it. I love how you add all the history in. My question's about the chemical reactions. Do you need to do that when the specimen is still fresh, or will it also work on a dried specimen?
Well, I've tried the ferrous sulfate reaction on dried specimens and it doesn't work. Some people are also doing a test with GAIAC, which I don't do because it doesn't seem to me to be terribly reliable. But the chemical tests have got to be done on fresh material.
Okay.
The chemicals that you use as stains, like Melzer's solution and so on, can be done on the dried material and they still work.
Mm-hmm. What's GAIAC, if I can ask?
What is GAIAC? Gum guaiac is a product of a Brazilian tree, I think. It's a stain which is used. You get a number of reactions of different colors. Somewhere in this room, I have a dictionary of chemicals used in mycology that would tell you more if you really want to know. I can probably find it in due course.
Yeah, I've never heard of it. So, okay, I've written it down and I'll look it up. Thank you.
We also have a thank you from Warwick. Warwick said that was so edifying. Thank you, Patrick.
Can I ask a question?
Please go ahead.
Yep. Patrick, you were talking back in the time of Russulaceae about using spore print colour in identifying. Are russulas not all white spored?
No. If you saw Russulaceae's spore colour chart, they go from white. They're all what Fries would have described as light colours, but they are never pink, they are never clay brown, they are never rust coloured, they're never black, they're never green. So they're in that range between white at one end and a sort of medium ochre at the other.
Okay. Okay. And another question, you were talking about the Chelocystidia and Pleurocystidia and Dermatocystidia. Do some species have them and other species not have any at all, or do they all have Chelocystidia, or do they all have some of them?
It's complicated. Not all have Dermatocystidia.
Yep.
So the red russulas, like the type of the genus Emetica, and some of our red russulas here have very clear Dermatocystidia and lots of them. And some russulas have none at all. Most russulas have both Chelocystidia and Pleurocystidia, but the shapes of them differ quite dramatically between different species, and the numbers of them differ too, so that some species can have Chelocystidia all the way along the bottom of the gill, so there are no Basidia on the bottom of the gill at all, whereas others have a mixture.
Yep.
So there is a big variation, and
Yep.
most of the European texts record this now, and most of the modern Australian descriptions do as well.
Okay, thank you.
Excellent. Thank you, Patrick. And also a thank you from Peter Boronsky saying that was fascinating. Tammy has asked a question. She said, I found Russula's disturbed cleared areas, and so wondering what trees may be associated with them. How far away might the Russula grow from a tree?
How far do the roots of a tree grow? It's extremely difficult to know, but there have in forests of a single species, it has been known to find that Russula fruit bodies have been found about 100 meters from the trunk of the tree. So that's a good distance, because you've got to bear in mind that the fungus itself, the mycelium in the soil, travels a fairly long distance, and it doesn't necessarily have to fruit next to the tree. Although generally, you see in some trees, there's a succession of fungi that fruit. There's fungi associated with a young tree, with a middle-aged tree and an old tree. And the fungi that fruit with the old tree fruit further away. The assumption has always been that the roots of the tree have grown further, and indeed they have, but also the fungus has travelled further in order to find nutrients to exchange with the tree. So it's some of one and some of the other that you're experiencing.
Very good. Thank you, Patrick. Do we have any additional questions?
No one's asked me whether you can eat them. You're all being very well behaved members of the QMS.
Yes, we're reversed to edibility, but I noticed that what is surprising to people in the field is often that we taste the lactarious milk, but the Russulas also, I notice.
You taste the taste test that has been used right from the beginning in Russulas involves taking a very small bit of the gill and putting it in your mouth, chewing it, and then tasting it on the end of your tongue. You can't be poisoned by it. You've got to know it's a Russula. And sometimes
it's a Russula, it's not toxic.
Well, if you ate the first Russula I showed you a picture of, if you ate a lot of Russula emetica, you'd get pretty sick. But tasting one little bit of a gill on the end of your tongue is just going to tell you it's very hot, and then you spit it out.
Very good. We have another question. Linda Reinhold is asking, can one individual fungus be attached to several different species of tree?
Oh, there's a difficult question. Can one individual fungus be attached to several trees? The answer is definitively yes. There's some really good work by an academic in British Columbia, which is reported in Merlin Sheldrake's book, published about 2 years ago, which deals with the connections between fungi and trees and how fungi actually can transfer nutrients from one tree to another tree. Does it go into species? I suspect not, but I don't know the answer, and I'm not sure that anybody else knows the answer either. Certainly, if you had a fungus that was that could coexist with, for example, birch and spruce trees, you find such Russulas in Sweden. There's no reason why it couldn't transfer nutrients or chemicals from one to the other. But whether it would do it more widely, I would very much doubt. I think there's a lot of specificity in Russula, as I suspect there is in Lactarius too.
Thank you, Patrick. I have another question from Penelope Sheridan, and she's asking, are there russulas with known economic value?
Yes, is the answer. Russulas are widely collected and sold in markets in Northern Europe as food. So if you go to the Baltic states, to Estonia, Lithuania, you go to Sweden, Finland, Norway, Denmark, Northern Germany. In all those countries, you will find russulas for sale in markets during the season. In Spain, Lactarius and Russula species are sold in markets, particularly in Catalonia, in Barcelona, at quite high prices. And there are people in New Zealand growing Lactarius deliciosus. They've specifically inoculated their pine trees in order to sell them. In Australia, I don't know of a russula that's sold, but Lactarius deliciosus is collected here and sold in markets, particularly in Melbourne and Sydney. I've never seen it in Brisbane, but then the fungus only grows in one forest in Queensland, and I'm not telling you where it is.
Very good. We have a mention here that someone, I can't determine their name, was saying that their parents are from Poland, talking about growing up eating russulas all the time. Some had to be cooked with milk to be edible.
I've heard that too, but I don't know. And there are fungi that...
That's Ola Roman, sorry.
Sorry?
That's Ola Roman has asked the question.
Yeah, it's certainly true that some fungi are cooked in milk and some fungi are boiled in salted water for preservation, but that also removes some of the substances, some of the chemicals that might make you sick. But it's rather like there are certain fruits that Aboriginal people eat that have to be treated in a particular way before they become edible. And the same is true with fungi. So for those countries in Europe which have had a fairly stable population over centuries and have collected fungi as food in the autumn, then they've developed such methods. I mean, Gyromitra is the critical example that everyone says it's poisonous. Every book you read says it's poisonous. But in Finland, in northern Finland, they collect them and they treat them in a particular way that renders them harmless and produce a very nice soup, which they feed you at the Finnish foray.
Very good. David Holden was mentioning there is a shop in Graceville that sells Lactarius deliciosus in season, so one to keep an eye out for, I guess. And Ola was mentioning that her parents said they don't know how to identify them, but their grandparents had the knowledge. So I think that's unfortunate too, that in the Indigenous community, sometimes the knowledge was handed down verbally, so it may have been lost.
My advice from my experience in Europe is that the best way to learn about fungi is from your granny, not from a taxonomist.
Very good.
That is if you want to go out and collect them to eat.
Are there any other questions that anyone had? No? Okay, so I might stop recording then.